All right, here we go. Chapter 17, section 2, part 1. So we talked in class that nitrogen can't be stored like glucose in glycogen, right? Because that ammonia is toxic. And so our nitrogen is lost as a result of protein and nucleic acid degradation. We have to replace it. We have to replace it with our diet, right? And so this degradation occurs all the time, all the time. And typically most people have a nitrogen balance, which means that their daily intake of nitrogen equals their excreted amount of nitrogen, right? So that's usually about 60 grams of protein per day that you're going to consume, right? But children and pregnant women are different, right? They have a positive nitrogen balance. And you got to think about, why do they need a positive nitrogen balance? Well, because they're undergoing tissue growth, right? They're still growing. So they don't just need to replace, they need more. And you can also have a negative nitrogen balance. A negative nitrogen balance is when you're in a disease state, like starvation, right? And then you don't have enough nitrogen. Either you're not consuming it or your body is missing the enzymes in order to metabolize it. So let's look at an overall big picture, right? There are two different ways that you're going to get amino acids. Either you're going to degrade the cellular proteins that you have, or you're going to eat protein and then you're going to degrade them. But either way, you have to degrade them. So typically this happens in a ubiquitin proteasome pathway or a lysosome pathway. And we'll talk about each one in turn. Now that we have the amino acids can then be recycled, right? So we can take those nitrogen containing groups and we can use them to make amino acids, to make nucleotides, and make biological molecules. We could also get rid of the nitrogen, right? We could export it out as waste, as urea. But in order to do that, we have to break it off of its carbon backbone. So if we break it off of the carbon backbone, the nitrogen group can go and be excreted as the ammonium ion is transported from peripheral tissue to the liver. So we go to the liver where it's incorporated into urea and then excreted out. So that leaves us with our carbon backbone, our carbon skeleton. It's some kind of alpha keto acid, right? And that alpha keto acid is used as a metabolite in lots of different energy conversion pathways, like citrate cycle, like gluconeogenesis, like we're going to make fatty acids, ketone bodies, whatever it is. So let's talk about the degradation of dietary proteins first. So what happens when you eat, right? Food's going to enter through your esophagus, and that stimulates the release of the hormone called gastrin. What does gastrin do? Gastrin has a couple of different effects. It's going to stimulate secretion of hydrochloric acid. If we stimulate secretion of hydrochloric acid into the stomach, that's going to reduce the pH of the stomach to about 2, right? It's also going to stimulate release of what's called pepsinogen. Pepsinogen is a zymogen. Remember, zymogens are those precursors to actual enzymes that will break down proteins. So we got a really low pH. That really low pH is going to denature our dietary proteins. It's going to kill bacteria, which is great. And then it is going to induce auto-cleavage, cleavage of that zymogen, that precursor, right? And so once we do that, then we're going to make the active form. The active form is called pepsin. And pepsin is going to cleave at those aromatic acid residues, right? So phenylalanine, tryptophan, tyrosine, right? All of those. Okay, so we've stimulated it. We've got things churning in the stomach. The things that are churning in the stomach kind of stay there for a while because you have what's called a pyloric sphincter, and the pyloric sphincter keeps the bottom of the stomach basically closed until you're ready to send that material down into the small intestines. So once that pyloric sphincter releases and you have what's now called chyme. Chyme is really a really acidic slurry, kind of the consistency of melted ice cream. right? Goes through the pyloric valve into the small intestines, and that first part of the small intestines is the duodenum. And so once the food hits that duodenum, you're going to get two hormones that are released. You're going to release secretin and the cholecystokinin. And then you're also going to release an enteropeptidase. An enteropeptidase, this is going to activate proteolytic pancreatic zymogen. So it's a cleavage cascade event. And I'm going to talk about that in a minute. But we'll make trypsin, we'll make chemotrypsin, all that kind of stuff. But the secretin is really interesting too, because that is going to stimulate the pancreas to make bicarb, right? And why is that important? The bicarb is really important to neutralize that chyme. Remember, we said the chyme was really, really acidic. And so if we release bicarb, we'll get that solution back to a pH of about 7. So then once we have the food in the rest of the small intestine, it'll go through the intestinal cells and then into the bloodstream. So let's look at that enzyme activation that happens during digestion. Remember, we have chyme, and the chyme enters the duodenum. And then one of the things that gets released as that chyme enters the duodenum is enteropeptidase. The enteropeptidase will actually cleave the zymogen trypsinogen, right? Trypsinogen will become an active enzyme, can then go on and induce more autocleavage, or it can go in and cleave other zymogens. So all of these that I've circled here, this one, this one, this one, they're all zymogens. So then trypsin can go in and cleave the rest of them. So now we get chemotrypsin, elastase, carboxypeptidase, all the things that break down those large proteins into little peptides and amino acids, which then can be transferred to the intestinal epithelial cells and finally into our blood. Yay. Okay, so what happens to proteins that are inside the cell? How do we degrade proteins that are inside of the cell? Well, this is another source of free amino acids, right? And it occurs continually in all cells. And you got to think that some proteins you want to have a relatively long half-life, and some you want to be short. So some are hours long and some are minutes long. But you need to have turnover of the proteins so that when you have different cell signaling events, the cell can respond. Because if everything just sticks around, you're never going to be able to change anything. And you also have to think, right? So you have to break down all those. Then you have to think that the cell's not always going to get it right when it makes proteins. So you're going to have misfolded proteins, truncated proteins, all that kind of stuff that you need to handle. So there are two pathways for, we'll say, cellular protein amino acid degradation, right? One is ATP independent and one is ATP dependent. The ATP independent process is non-selective. The ATP dependent is selective, right? So the non-selective pathway is just to put everything into a lysosome and to put a really low pH, about pH of 5, to break down and degrade the proteins. So it doesn't require ATP for that degradation to occur. You have some proteases in there that are going to help with that, but it's ATP independent. Now, you do have to keep in mind that if you want something that's at a pH of 5 and the cell's normally 7, you're going to have to use an ATPase to actually pump hydrogen ions into the vesicle. So the vesicle's derived from the Golgi, and you do have to consume ATP to pump hydrogen ions in there, but once it's in, you're all done. So now we have these proteolytic enzymes inside of this vesicle. We don't want it to burst open, but if it does, it's okay because those proteolytic enzymes only work at really low pHs, so we're not too worried about leaking. The second method is really selective. It's ATP dependent, and it degrades proteins that contain ubiquitin polymers on the end of them, and it feeds them into a proteasome. So this is very, very selective. So it only occurs to proteins that are ubiquitinated. And it goes into a proteasome that is composed of 3 big subunits, right? You have these yellow ones, these yellow caps on the ends. These are the 19S subunits. And then you have a central 20S core. Now the S just stands for Svedberg units and their sedimentation rates. But the 19S are really regulatory. So they control entrance and exit of proteins, right? They also, the 19S is the part of it that binds to the ubiquitinated protein to make sure that only those are fed through the proteasome. It also helps to unfold the protein. And then once the protein is fed into it, right, and it gets to the catalytic core, the catalytic core starts cutting it up. Okay, so ubiquitin is for dietary proteins and for cellular proteins, but it's just things that are intracellular. So from this, we're going to get amino acids that can either be recycled and used for something else, or they can be deaminated and we can keep their carbon skeleton as that alpha keto acid and then remove that nitrogen as a waste product, right? So these proteins that are tagged for degradation are tagged on the lysine residues with ubiquitin. Now, ubiquitin itself is pretty long. It's 76 amino acids long. And it has 7 lysine residues inside of it, right? And so these ubiquitin molecules are linked together, and we basically represent them in these as like a yellow triangle. You'll see them. So we don't really show the structure. We just say, hey, that's a ubiquitin. Okay. So let's look at these different reactions that occur in order for ubiquitin to ubiquitinate your target proteins. So we really need 3 classes of enzymes in order to make this happen. They're called the E1, E2, and E3 enzymes. So basically, E1 is going to attach a ubiquitin molecule to E2. Once you have E2, E2 is going to attach the ubiquitin to whatever your target is. And they say TP, I believe, is their target protein. Yeah, there's your target protein. And then E3 is going to facilitate ubiquitin of the target proteins by forming a complex. So we call this one the ubiquitin ligase, ligase. And so this is the one that is really going to form the complex between the target protein and the ubiquitin. So we're going to walk through all of these steps. But one thing that's really important is to think about how many different genes of each of these classes of enzymes there are. The E1, there's only 2 genes. And really, that's okay because the only job of E1 is to attach ubiquitin to E2. So it's not a really big job. Then E2 is the one that's going to bind. It actually binds to E3, and it has the ubiquitin. And then E3 is actually going to be able to transfer that ubiquitin molecule to the target. So we actually have 30 different E2 enzymes. But here's where it gets crazy. At E3, we have 500 genes encoded in the human genome for E3 ubiquitin ligases. And so these have to be very, very specific for their targets so that you're degrading the right thing. Very, very important. Okay. So if you look at our first step, right, in an ATP-dependent fashion, we're going to ubiquitinate our first E1 enzyme. So once E1 is ubiquitinated, basically what we're going to do is we're going to take this ubiquitin, and we're going to transfer it onto the E2 molecule. Okay, so now we have an E2 molecule that is ubiquitinated. Once E2 is ubiquitinated, it is going to bind to an E3 molecule. And you can see on this E3, you have 2 binding sites. The first binding site is for E2 that has a ubiquitin attached to it. The second binding site is for your target protein. There are only certain target proteins that will bind to the E3 complex. But once you have both things attached to E3, then you're going to have a transfer of ubiquitin from E2. So we're going to take it here from E2, and we're going to add it to our target protein. So once we do that, the first go round, we only have one ubiquitin molecule attached. So we're actually going to kick out this E2, pick up a new E2 that has a ubiquitin already attached to it, and then transfer it. So we end up with a molecule that has at least one, two, three, 4 ubiquitin molecules, because it must have at least 4 ubiquitins attached in order to be targeted for degradation. So how do you say this protein should be ubiquitinated? This protein needs to be degraded. Well, there are a couple of different things. And half of them are dependent upon the target molecule, and the other half are dependent upon the E3 ligase. So typically we look at the target, and if the target protein loses its N-terminal methionine residue, then that is a protein that needs to be degraded. And so that is going to be, that target protein is going to be delivered to E3, ubiquitinated, and then destroyed. Also on a target protein, right, always has to be phosphorylation. If you phosphorylate a particular residue in the target protein, then that target protein will go to the E3 ligase, get ubiquitinated, be degraded. The other way that you can regulate this is through the E3 complex. You can actually phosphorylate the E3 complex to make it more active or more receptive to a certain target protein. So induce a conformational change where this target protein will fit better in the enzymatic pocket and therefore get ubiquitinated and degraded. The last way that you can alter the E3 complex is that you can actually have an activator, an additional protein that will bind to E3, again, induce conformational changes, changes the affinity of E3 for the target protein, or E2 really, to facilitate ubiquitination and degradation. Okay. Oh, and then the last one. So we said you could change your target protein. That was the first thing we talked about. The second one we talked about was biochemical changes in the E3 ligase, right? So the last one is called the end rule of protein degradation. So if you have particular N-terminal amino acid residues, this is going to signal these target proteins to be degraded. And these are proteins that you want to have a really short half-life in the cell. You don't want them to hang out a long time. You want them to be ubiquitinated and degraded relatively quickly. So usually it's ones that lack methionine on the end. Instead, they have a phenylalanine or a leucine or a lysine, whatever it is. But those are the ones that are like transcription factors that you want to turn over very rapidly in response to whatever's going on in the cell. So make sure you go in and view the protein degradation animation. It's really cool. Animations are great. Okay, that's the end of part 1.